in an alternate universe, Sky High is a school for humans with superpowers. It is a school for socks with superpowers. Also, it's called Sock High, not Sky High. A long time ago, there was a scientist dude who went mad. He abducted families of socks to be used for his experiment. They were able to arrest the scientist, but those socks were never the same again. They acquired weird abilities, like bugs stayed away from them, and they breathed underwater. All the other sock people were so weirded out that they shunned the poor socks. They were nicknamed GMOs, which didn't really stand for anything, but it instilled fear and awe from those who heard the title, so they stuck with it. The GMO socks were shown, but because of software, they were still required to have an education. So Sock High was born. In Sock High, we studied all the regular subjects almost socks did, exception of gemology. In this subject, they would study about how they came to be and what the purpose of the scientist was. It was theorized that the real purpose of the scientist was to improve sock race. If you think about it, all the abilities given to them were good things. A league of GMO students decided that they would fulfill the scientist's goal. They were gonna recreate the machine the scientists used, but a more flexible version. After months of hard and secret labor, their creation is finally finished. They called it the machine gun. They crept through the night with their guns, using their extra super abilities. They shot socks. Silencered. The next morning, millions of GMO socks look at their mirrors. Then they notice they have changed. They feel stronger, faster, better. Day by day, millions turn into GMOs with the help of the machine gun. Then a new race of sock people occupied planet Drawer, fulfilling the scientist's goal.
The gene gun. The technique of tissue culture is used in genetic engineering. In this method, tissue samples are taken from plants and masses of undifferentiated cells called callus are grown. The new genes are added to these cells. The goal of transformation is to get one or more copies of a new gene into the nucleus of a cell where the chromosomes are. One transformation method is the gene gun. This method uses microscopic gold particles to deliver the genes into the nucleus. Hundreds of gold particles are coated with thousands of copies of the new gene. A second gene that codes for an easily detectable trait, called a marker gene, may also be added to aid in determining which cells have been transformed. The coated gold particles are accelerated with air pressure and are shot at a pile of callus cells on a Petri plate. If the gold particle lands in the nucleus, the genes dissolve away from it, and may then incorporate into the DNA of the chromosomes. Each time a gene inserts into a chromosome, a different event is created. Events are determined by which gene is inserted and where the gene inserts itself. The same gene inserted into different locations can exhibit differences in expression. When a new gene inserts into a chromosome, the chromosome's DNA separates, allowing the new gene to insert without replacing any existing gene. It is similar to cutting in line. The gene is now part of the cell's DNA and will be passed on to the offspring.
There are a number of different methods out there to help us do transfection. Early on, a lot of people used calcium phosphate precipitate to transfect cells. You had to get the pH just right, and the transfections weren't that great. Then along came cationic lipid reagents and everything got much easier. Now you just mix the DNA or siRNA with the lipid and add it to your cells. Very easy. You mix the two, and the negatively charged nucleic acid molecules bind the cationic lipid and form a complex that can bind cells and be taken up through the endocytic pathway. Once in the endosome, the nucleic acid needs to escape into the cytoplasm before being degraded. This endosomal escape occurs by many different mechanisms, including membrane fusion and disruption by interactions, and lipid exchange between the transfection reagent and the endosomal membrane. Once the endosome is disrupted, the nucleic acid is free in the cytoplasm and available for nuclear shuttling in the case of DNA or binding the RISC complex in the case of siRNA. The lipid reagents have continued to evolve since they were introduced, and there are newer reagents available now that work on common cell types, as well as cells that used to be considered hard to transfect. You
don't realize it, but actually, there are a lot of biotech foods in the market.
There are tomatoes, eggplants, potatoes, corn, rice, milk, papayas, soybeans, and salmon.
Now you may be wondering, I don't know any transgenic agricultural animals that have been bred to date. What are some examples? And also, how are they different from their unaltered counterpart? Okay, one example is a cow. Before it becomes a genetically modified cow, it can only produce, let's say, 1 carton of milk. But after it becomes a genetically modified cow, it can produce up to 4 or something. Actually, it just produces more. Yay! Next one. And then, this is a sheep. Before it becomes a genetically modified sheep, it has this much wool. But after it has this much wool, which is more than the original wool. Yay! Another example is a salmon. This is a salmon. Both at 18 months, this regularly developed salmon is weak and sad. But compared to this super developed salmon, which is developed faster more than the undeveloped salmon, yay, awesome, right? Okay, next. And lastly, this is a chicken. So it's also sort of like the salmon where it gets developed faster. So in due time, let's say 2 years, while your lame neighbor has 1 chicken, you have 2 chickens because it developed faster. Yay!
What are the advantages of using DNA technology in agriculture? This technology of manipulating the DNA provides scientists much more control over the qualities of the crops and animals. Favorable traits may be expressed by an organism so that it can be more competitive. A higher amount of yield can be produced. Resistance against pests, viruses, and diseases can be incorporated. The flavor can be enhanced. There can be an increase in the nutritional content of the food. Believe it or not, there is a great chance that a part of the food we have consumed today is a genetically modified organism, or a GMO. However, this is not known to the public because the food doesn't really get a tag on it that marks it out as a GMO. Although GMOs are developed in order to improve its quality, because it is artificial and has been tampered with by science, the public generally disapproves of GMOs. This may be because they think that anything against the natural state of things is unacceptable. Another reason may be that they simply are afraid of change. GMOs are recent discoveries, and this has not allowed enough time for it to be tried and tested over and over again, especially since people are doubtful of their safety. There is also the concern of the potential effects of releasing these GMOs into the wild. Although only a small part of their genome has been modified, there is still a possibility that it will affect other characteristics that will render the GMO harmful in the ecosystem.